Hi there, I'm Tim, President, CEO, and co-founder of Forge Biologics, and it's my great pleasure to stand up here and talk to many of our competitors and friends that are here in the audience today. So one of the fun things about this particular conference is just noticing to see how much manufacturing has really become such an integral part of many of our daily lives and our careers. So happy to share with you a bit more about today's story. And I'm Tim Millard. I've been in the gene and cell therapy industry since the late 90s, going way back to cystic fibrosis gene therapy, and have formed a number of different companies, built great teams, and have the privilege to work with a lot of great colleagues, and hopefully some of you again in the future. So one of the more interesting things that happened to Forge, so Forge was founded in 2020, and we were actually acquired at the end of last year, and we completed the acquisition by Ajinomoto. So Forge is now part of the Ajinomoto group, and probably one of the number one questions that I get is, hey Tim, how has this gone? How has the integration been? And it's been actually fantastic. One of the nice things about Ajinomoto is that the company's been around for over 120 years, and they have a long-term vision, so we're already planning towards Roadmap 2030. So if you look online and you hear a lot of things about Ajinomoto and Forge, you'll get to see some of that. But they also have decades-long commercial infrastructure, which is certainly helping us as we think about our global expansion, and just continuing to help invest into Forge and to continuing to build out our facility. So it's been a fantastic journey so far with them, and looking forward. Their main thing has been, Tim, we're here to help make sure that you guys keep doing what you're doing, so hope to provide a lot of confidence to our clients. So Forge is a leading global genetic medicine contract developer manufacturing organization. Just a little bit by the numbers, we have over 20 GMP suites with AAV and full GMP plasmid production. It's been fascinating to watch a lot of things in the industry about some of how plasmid manufacturers seem to have taken a lot of focus. We have the ability to make over 200,000 liters capacity and greater than 300 employees and continuing to grow. In 2023, we had over 10,000 applications into the company to hire about 150 positions. So in a landscape where many, many biotech companies were letting go of employees, we were continuing to build, and happy to do so. So we're going to talk a little bit about how we're improving accessibility to genetic medicine. It's really through better manufacturing and expertise. One of the fascinating things that I think our team gets exposed to is when many clients, and we are about to sign our 50th client, as clients bring in their plasmids, often coming in from academic facilities, one of the things that we find a lot of the times is that we see flipped ITRs, or we see that things are overstuffed and overpackaged. So helping with molecular improvement, which leads us to be able to help improve their process. And we do have a platform process that's been, I think, great uptake by clients, and that includes a lot of enhanced analytics. So trying to help move things forward for every program, as they scale up or scale out, and doing a lot of integrated regulatory support. So our regulatory team has sat on a number of pre-IND, IND calls, working with scientific advice meetings, and now moving around into the PMDA and other groups from a regulatory perspective around the world. We also have a strong R&D element at Forge, where we continue to discover and look at novel ways to help improve and enhance productivity and efficacy for gene therapies. So lots of text, a little bit small. I think the punchline that I want you to take away with is we discovered, that certain elements are required for AAV production, in particular the L4 region of the 33K protein. So this was published recently, but you need this to actually make and improve AAV production and manufacturing. So this was actually published in Human Gene Therapy in the past few months. So take a look at it. If you're having problems with some of your AAV production, you may actually want to look at this element, that's required for efficient and scaled up AAV manufacturing. So really what this is just to help demonstrate is that the Forge team continues to try to be innovative, but also to push the science forward. We're not only manufacturers. We do try to start with the end in mind, however. So starting with a commercially viable bespoke platform helps support easier development in your scale up. So, when we start, you can come right into the beginning and start with our research grade Blaze offering. This offers full traceability as you're able to go forward. Our cell lines were one of the most traceable cell lines in the industry right now, but fully commercializable, animal component free, and all single use. So this allows everyone to adapt to a particular client needs, but as you move forward from anywhere from 250 mils all the way up through 5,000, up to 1,000 and 5,000, this actually allows you to use our platform. Our data master file is on file with the FDA and currently being filed with the Canadian and other regulatory agencies around the world. So you want to try to leverage a bespoke platform, right, as a foundation for development. And since we're here at the Alliance for Regenerative Medicine, many of you are familiar with this type of manufacturing process, but you're really breaking this down into upstream and downstream process. Forge has expertise in both areas and able to help you actually work on additional ways to scale up your program. So we're also trying to do this coupled with client-specific PD, right? So every client's program is just a little bit different, and while every program is bespoke, you are able to use a platform to kind of use as a jumping point. So commonly requested studies, affinity chromatography, there's lots of downstream evaluations. We've also been really focusing more recently on optimization of transfection parameters. So, and you can see here, just using one client in particular, we're able to scale their process over almost 900-fold. So again, when you take the time to realize the manufacturing efficiencies, this can significantly impact your program success. So be cautious about any of the programs where you jump in and you're like, great, we can use your platform. You are always going to want to do some level of engineering runs. It saves you time, it saves you money. Just be careful about trying to jump to the end. We have over 20 assays in-house for efficient and accelerated characterization and release. So, we try to do everything in-house, where we're trying to do both safety and quality, concentration and potency, do all the integrity and identity and composition testing, and purity. So we've tried to move as many of these in-house so you can start with doing plasmid production, do your full AAV GMP, and have many of your release qualifications and assays done in-house. So something that's fun, and one of the things you'll read about a little bit more later on this year is just how NGS is really taking a hold, and we've brought that all in-house as well. With a spotlight, however, on comprehensive characterization, we really wanted to go through a hybrid sequencing approach, right? So we'll focus just on Forge's hybrid approach, so we can do short and long read sequencing. Coverage depth is very, very high compared to what else is out there for other sequencing methods. It's orthogonal, so there's no gaps in coverage, and our throughput is pretty high due to in-house bioinformatics. So from an application perspective, being able to comprehensively characterize your plasmid and combination AAV products, really it helps because it'll help detect any contaminants very, very early. And one of the things that every group is really trying to do is how you do automation. And as the field has continued to move forward, it's nice to be able to see robots coming in and actually doing the work, and I am supportive of our new overlords. So, but as we think about multi-parallel bioreactors, we do have TCAN liquid handlers, and again, as I mentioned, we do have next-gen sequences. So this helps improve data integrity, which from a compliance perspective, definitely enabling faster analytics. So again, you can come and you can watch these work, important. Our process development and our GMP scale-up enable a downstream commercial suspension platform. So from here, these are all bioreactors that are in-house in our Columbus facility, anywhere from a very, very small amount of 100 to 250 mils, all the way up to 5,000 liters. And we're looking forward to talking more about the 5,000 liters in the upcoming months. But today, you can come in, you could do a 50-liter research-grade production run. You can use that to jump into 200 liters, 500 liters, and 1,000 liters for GMP. And again, all of this is single use, which is really, really important when looking at your downstream development. What's our facility look like? Well, this is a nice map of the facility. Again, we have infrastructure to be able to support close to 450 employees. We have all of our infrastructure under roof, GMP warehouse, 25,000 square feet. We have upstream and downstream process development suites. And we have future expansion space. So we'll have some fun announcements coming out later this year about some additional programs and additional platforms that Forge is rolling out for clients. And talking about fill, right, it seems to be a nice bottleneck, but we do have automated operations from the Cytiva microcell. So this is aseptic filling with full automation. It's an ISO 5 gloveless isolator. being able to do batches from about 1,000 or 100 to 1,200 units every 8 hours, and it's ready to fill, so it's good for commercial and different container closure types. We also have semi-automated, the Watson-Marlow, right? So semi-automated, we can do about 500 vials in 8 hours. You can do this with a repeater pipette and manual crimpers and an ISO 5 or grade A biosafety cabinet. So custom fills anywhere from 0.2 to 20 mils. Regulatory. So as everyone I think knows, 70 to 80% of the regulatory challenges in today's day and age in gene and cell therapy come from regulatory. So we're able to reference an AAV platform, both for plasmid and AAV master files. We do sit on many regulatory calls with our clients, and trying to participate in as many meetings as we can to help them. Right now, we've sat in what seems like a lot of U.S. and pre-CTA meetings in ex-U.S. regions, and coming up in 2024 is more and more APAC. So, and extensive industry experience from the regulatory team supporting many of those products, greater than 35 years in all phases of development. And as Ajinomoto has come in and really helped accelerate our expansion into Europe. So we're establishing a client development presence in Europe. We have offices in Madrid, Spain. Our next stage of development currently meets and will meet EMA classification requirements. Our filling suite and shipping offerings offer for global cold chain supply. And we do have QP declaration for part of our facility, so you can come and actually make stuff at Forge and use it in Europe. And again, as we're, Forge's team is completing CTAs with our new EU group. Currently serve over 45 clients, again, almost about to sign our 50th client. 95% of our clients start their production using our Ignition HEK293 suspension cell line, and greater than 80% of our clients use Forge's proprietary P-Ember ad helper plasmid, and we have new versions of that coming out again soon. So with a broad range of client segmentation, we see this all from private biotech, public biotech, academic, foundations, and we've made pretty much every capsid under the sun, including many novel capsids. So, and just to announce a call out to Ray, one of our really excellent clients that we've publicly announced, as well as Solid Life Science and New Hope Foundation. So just really over the past 18 months, we've successfully managed or manufactured almost 300 batches across many serotypes, from Blaze process development and engineering runs and up to GMP. So we've been very, very busy for the past 18 months alone in our manufacturing suites. And again, that's up to 1,000 liters. So when we think about how you scale and starting to try to meet commercial demand, 1,000 liters are where it's really one of our main workhorses. And so with that, I'll say thank you. The Forge team is here, and if you have any interest to meet with us, we have a booth around the corner and some private offices as well. So thank you very much.